All right. Well, I think now we can go ahead and get started. So we're going to have two speakers today. Our first speaker will be Paulina Pais. Paulina is a bioinformatics research associate in Ryan Corse's lab. Paulina earned her bachelor's in computational neuroscience from UPenn, and prior to Gladstone, she worked for biotech companies Octant Bio and Loyal for dogs, as well as at the Rockefeller University. So Paulina, welcome.
Thank you, Santi. Is this when I share a fun fact about myself?
Oh, yeah, and please share a fun fact about yourself.
Awesome. So fun fact about myself, I'm from Guatemala, and during the weekends, we like to go hiking volcanoes. So that's like a typical weekend activity in Guatemala. Today, I will talk to you about how we are facilitating broad data sharing of single nucleus ATAC-seq data through a flexible online portal. So the Courses Lab is interested in the epigenetic factors influencing neurodegenerative diseases. And one of the tools we use to study this is called ATAC-seq. If you're not familiar with ATAC-seq, it stands for Assay for Transposase Accessible Chromatin Using Sequencing. Today, I won't have time to go over the molecular background behind this, but I can point you to a publication that was released by our lab that goes into the protocol in detail. For the purposes of this presentation, it's suffice to know that ATAC-seq identifies regions of accessible chromatin, often bound by transcription factors. And the way we like to think about it is that it sort of shines a flashlight on the regulatory elements that bind to the DNA. And that signal is what we call a peak. With that data, we can do all kinds of things, including transcription factor prediction, transcription factor footprinting, and identifying allelic accessibility. So there are a couple computational tools out there that help biologists make sense of single-cell ATAC-seq data. ArchR was developed by the head of the Courses Lab, Ryan Corses, and his colleagues when he was a postdoc at Stanford. And I might be biased, but it is the best by far out there. Here I'm showing a bird's eye view of the end-to-end workflow, and what I hope you can appreciate is just how comprehensive it is. A key feature of ArchR is the interactive genome browser. Scientists find it really useful because sharing and visualizing ATAC-seq data is very challenging for various reasons. One of them is that it's time consuming. Another is that there are multiple software tools required, which make you group single-cell ATAC-seq fragments in a way that sort of counteracts the whole point of doing single-cell assays. They're also not dynamic because any change you want to do to the cellular groups or adding samples, for example, requires regeneration of bigwig files. So what we found, the typical use case was that people found a way around it by just piling lots of static PDFs and just creating a mess out of it. And so one way that the developers of ArchR tried to facilitate this data sharing was with the ArchR Browser, which is already included in ArchR today. And the ArchR Browser is basically an interactive genome browser that can be launched with a single line of code. Here's a screenshot. It is based on Shiny, which is an R package that makes it easy to build interactive web applications. And it is possible thanks to a data storage strategy used by ArchR, which I will explain later, but it's called arrow files. And this enables real-time track level visualizations. However, it is hosted locally, and so to reproduce the app, scientists would have to obtain all the files and objects, which if you have tried sharing sequencing data, you might know how much of a pain this is. And so our goal was to build a plug and play Shiny application for ArchR users. And that could be exported and shared at the time of publication. The implications of this were that we had to write a function, we would call it export Shiny ArchR, that automatically generates a set of files specifically based on user configurations and data that would be required to deploy the application. And while this sounds easy, just creating one function, in reality, there's a lot that went behind it. And so this is what I want to share with you today. So ArchR's file storage is based on the rapid access HDF5 format. HDF5 stands for hierarchical data format and was designed at the National Center for Supercomputing Applications. These types of files contain various compression filters that allow us to store, access, and organize large amounts of data. Here I added a link if you want to learn more about them. I think it's very smart the way that they put this together. But for today, all you need to know is that they contain everything we need to launch the browser. However, you'll see later on, I will explain why it wasn't just as easy to deploy them in the server and just share these files. So there are many ways to host Shiny applications, and we looked into many of them, from AWS, so Amazon Web Services, to mounting your own server, which is honestly not that complicated, but it does take time. And we know that scientists, that's the scarcest resource they have. So we really wanted to go all the way and understand where users could host their applications in a way that was easy and free and widely used. So shinyapps.io is an online service for hosting Shiny applications in the cloud. And it has many pros, including the fact that it's widely used already in the community, so people know about it. It interfaces with R, the language that ArchR is in. So then it would be relatively easy to integrate into ArchR. It also has a free tier and a small learning curve, and it's user-friendly. However, it has two big cons. One of them is that they have a speed and memory file limit, which could technically be solved using the HDF5 arrow files because they're easy to access and they have their own file system. But the drawback is that they only give you 1 GB for your largest instance in the free tier. And we didn't want our users to have to pay to maintain an app. But the arrow files that I talked about, the HDF5 files, are 2 GB, around 2 GB for the test ArchR project that we had, which was also only 10,000 cells. And we know that people have projects that have 1 million plus cells. And so we knew that we could not put these HDF5 files into shinyapps.io. So we had to find another way to compress them. And we could not call the National Center for Supercomputing to help us. And so instead of using the HDF5 files, we read about this other form of data compression called run length encodings, which stores repeating consecutive values as one entry. And so as you can see here, what you have is the length mapped to the value. So for example, 2 only appears once, and so the length of that would be 1. Then 3 appears twice, and so the length of that would be 2, and so on. RLEs are used in G ranges, one of the most popular packages for representing and manipulating genomic intervals. And to create them and reflect what ArchR sequencing data had, what I did was calculate insertions from fragments, then bin regions of interest, and overlap insertions with bins. And so this is a function within a function that would feed this data to the export Shiny ArchR function and pre-compute it before we export the app. We found that this worked really well for the test ArchR project that we had. It was only a couple megabytes, but I also wanted to see how it scaled the more fragments each cluster had and the more cells in each cluster. So to the left, you can see a plot of on X, the number of fragments in the cluster, and on Y, the size of the coverage RLE objects. Then on your right, you can see the number of cells in the cluster mapped to the size of the coverage RLE object in megabytes. And so even though both have an upward trend, what I hope you can appreciate is that there are only a couple megabytes compared to the 2 gigabytes that one arrow file was. And so we were very happy to see this. After some modifications and finishing the functions, working on the UI side of the application, this is a screenshot of what the ArchR browser looks like, except this one is created using the run length encodings instead of the arrow files that I mentioned. And so we can keep the data intact and also have it in a low memory footprint that can be exported to shinyapps.io. And here's the link if you want to see later an example of the application. But in addition to just mapping the peaks and co-accessibility and genes, we also wanted to add another type of visualization that is very helpful for sequencing data, which is called Umaps. UMAP, if you're not familiar with them, stands for Uniform Manifold Approximation and Projection. They're very useful for dimensionality reduction and sort of stratifying, understanding what kinds of cell types you have and what's in your data. So in the case of ArchR, UMAPs are generated per matrix of interest. The main ones for ArchR are 3: 1, gene scores, 2, gene integration, and 3, motifs. Per each of these matrices, you would have features of interest. So of course, for the first 2, the features of interest would be genes. For the last one, it would be motifs. There are many features of interest, as you know, genes 20,000. And so we wanted to be able to generate a UMAP per feature of interest. And it was not possible to pre-compute and store the matrices because since we have it's a combinatorial problem that basically results in a lot of UMAPs, we could not store that. And the processing time per this is 7 hours per matrix, which we know users would not want to wait for. And so this is just a screenshot of one of the first tests we did, where we tried to store the 3 matrices, and that was 1 GB in itself. So that's not even accounting for the browser that I showed before. We knew that we had to figure out another way to export this and host it on shinyapps.io. And so our solution to this was storing rasterized images in HDF5s. So yes, we're going back to HDF5s, but we did it in a clever way so that it wouldn't hold that much data and they wouldn't be as hefty as 2 gigabytes. So the way we do this is by writing a function that holds input weights in memory while plotting this embedding, the UMAP, to cut processing time. So instead of 7 hours, it's going to be 1 to 2 hours. Then we overlay rasters. So there's 2 kinds of images, rasters or vectorized images. And we know that scientists for publications and things like that, they need to modify their own images in Adobe Illustrator, things like that. And for that, they need vectorized images. So the axes are vectorized, and you just overlay these plots, the embedding, on top of the axis. And that way you can store more than 20,000, which we call native rasters. They're sort of like raw integer formats of what is contained here, the colors that are contained there in the HDF5. And then we store all of that in the HDF5. And then that way we keep inside the 7,000 file limit that shinyapps.io has, because otherwise, going back to this, it would be 20,000 just for one matrix. So it would be 40,000 per two. And if we add motifs, it would be like 41,000 files just for this. This approach, we were happy because it scales linearly with the number of genes instead of the number of cells. So one thing we had to keep in mind for all these solutions and limitations that we had with ArchR with shinyapps.io was that we wanted algorithms that scaled with the number of genes and not with the number of cells, because the number of genes is predefined. We know what the upper limit will be, versus the number of cells. It would be, our test projects would be like I mentioned, 10,000 cells, but we know there are people working with 1 million. So we wanted to be able to provide this app for those kinds of projects as well. And so the future directions are that in the next release of ArchR, we want to have this shareable data portal where users can visualize genome tracks, peaks, co-accessibility, and clusters, as well as UMAPs and motifs of interest, sort of like an ArchR Browser 2.0. We will also have updated documentation, and it will be containerized using two tools called Docker and Renv, which is a dependency package manager in R, which will make ArchR workflows reproducible. And that I won't talk to you about today because of time constraints, but I'm happy to discuss how we did this offline. And so I just want to say thank you to our lab members and to my collaborators, Ankush Sharma and Akshay Suhag, who have helped me. They're from Norway and from Switzerland, have helped me developing the app and understanding the how Shiny works. Thank you.